So this is a continuation of the last discussion, but we'll start with the digestive system. Now, the digestive system, I'm not going to go from top to bottom through this. You know about the anatomy of the system. You're going to find in your course pack 2 PowerPoints. They're both kind of long. The first one that just came after the arteries and vein chapter says at the top, anatomy of the digestive system. I'm not going to touch that one at all. That's just an overall review of the anatomy of the system. That should be 99% familiar to you from Biology 105. Where I'm going to pick up right now is in the second PowerPoint. So you're going to flip over a few more pages, and that brings me to what page? The second presentation, go all the way over. Ty, and where are we? Page? 308. Jump all the way over to page 308. Okay, so the pages you're skipping over are just the anatomy of the digestive system. Look over it, but don't spend a lot of time on that. Okay. Now, the digestive system. There's some really cool things about the digestive system, and I want to focus on some physiology of this system, not rehash all the anatomy. And so what you'll see here is that I am going to only cover a few slides. In other words, slides 16 to 97 about teeth and swallowing and the salivary glands and the stomach, I'm not going to cover. I'm also not going to talk about the small intestine, 133 to 154, or the large intestine, 187 to 204. But I will put a significant number. Here I put 15, I don't know, it's at least 15, maybe even more, extra credit points on the next exam. Okay, now so the extra credit questions will come from the areas I'm not going to discuss. So review them on your own, look through them. If you get them, great, they're extra credit. If you never spend a minute on it at all, it's not going to hurt your grade. Okay, it's only going to help your grade if you can find some time to review that. And honestly, the questions I'm going to ask you about that are quite basic. Okay, so that's coming your way, the extra points on exam 4. So last couple semesters, I've had people scoring well over 100% on this exam because of those bonus points. So, again, it's my gift to you. So what do we have coming down the pike? I'm getting a few concerned emails from students. So what do we have? Some of you have already looked at, or at least have seen, on Connect, there's an extra credit quiz. People who have done it have seen a nice boost in their grade. It's 10 points. That's 1% in your overall grade. At this point, it's almost 2% boost in your grade because we're only about halfway through the points. There'll be another quiz coming up. The first quiz is on the lymphatic system. The second quiz is on overall cellular metabolism. Again, topics that I'm not directly discussing, but they're open book, open resource. If you don't do them, it doesn't hurt you at all. It only helps you to do these quizzes. So I encourage everyone, even if you have only 5 minutes and you never anticipate doing it, at least go in and click A for everything and get something. Something's better than nothing. These are bonus. There will also be 14 overall lecture quizzes, only 10 of which are going to count. So in the end, I'm going to drop your 4 lowest lecture quizzes. Either you missed them or didn't do well. There's also going to be your second case study. The first case studies are graded. The scores are recorded. No one did poorly. A couple of you lost a few points for wacky or incomplete citations. And so, again, if you didn't get a perfect score if you didn't get at least a 25, come see me. And let's make sure you fix what little things were wrong with the case studies. There's also going to be your Connect assignments. If you've been doing those, you'll get 30 out of 30 at the end. And your lab reports, I have not graded those yet, but they should be strong as well. So there's a lot of grade boosting things coming your way, as I've said before. So keep looking for those. And here's another one that I haven't mentioned before, these extra credit points on exam 4. So let me start this conversation now. Again, this is that second lecture. And I'm going to start off with a discussion of the enteric nervous system. This is something you may never have heard about before. I did not discuss it at all when we were discussing the digestive system last semester. And that is, you have more neurons in your gut regulating your digestion around your esophagus, down around your stomach and your intestines, relating to the overall motility and secretion and blood flow to your gut than you do in your spinal cord. It's amazing. Over 100 million neurons are found, and these neurons are completely independent from your central nervous system. It's sort of like the heart. These neurons are independent. They are influenced by your central nervous system, right? But they do not have, they're completely independent of the central nervous system. So this is referred to as your enteric nervous system. And most of this are sensory neurons. And those sensory neurons, right, are measuring the motility, the stretch, the pressure that is on your gut wall. And that's sending signals back to your brain, and then your brain is helping to regulate this overall. Now, there are two sets of neurons in your enteric nervous system. The submucosal plexus, recall a plexus is a what? A group of neurons, right? A cluster of neurons that travel together. And these are also called the Meissner plexus. These are in the submucosa. Can you imagine where that is? If you think about any of your, like go through your stomach or your intestines, what are the layers? From the inside, the mucosal layer, that's the mucosa. Just underneath that, a blood vessel rich area is the submucosa. Remember this? And then there's the muscularis, the smooth muscle layers. Around the stomach, there are 3 layers of muscle. Around the rest of the intestines, there are 2. And then the outer layer is the serosa, remember that around the stomach and around your intestines, you have the visceral peritoneum, the serous membrane. So these submucosal or Meissner plexus neurons are in the submucosa. And what they're going to be doing is controlling glandular secretion and movement of what's called the muscularis mucosa. There is a little layer of muscle even within the mucosal layer, and that's going to help squeeze out the secretions. I never really talked about this before, but how is it that those little glands that secrete things are releasing their products? They're actually being squeezed a little bit by little muscle cells called the muscularis mucosa, so the muscle of the mucosa. Then there's a myenteric, also called the Auerbach plexus, and these are parasympathetic ganglia between your 2 layers of muscle. So remember, you have the muscularis, and that's a layer of circular muscle and a layer of longitudinal muscle. In between those 2 layers is the Auerbach or the myenteric muscle or plexus. Now look at that word, myenteric, muscle, myo, right? So the mesenteric plexus. And this is the layer that's going to control the peristalsis. Peristalsis, think of it as that rhythmic movement of the muscularis that pushes products throughout the gut. Another term we haven't talked too much about are the mesenteries. The mesenteries, what does mes mean? Middle, right? And entero, right? Entero, the what? The intestines, right? So mesenteries are what? In the middle of all the intestines, right? This layer, this very thin layer of connective tissue, almost like sheets, and they're loosely suspending the stomach and the intestines from the abdominal wall. If you think back to dissecting the fetal pigs, and if you were to lift up all of the intestines, you would note that they were sort of connected to one another through a very thin, almost translucent layer. Those are the mesenteries. And also these mesenteries are helping to suspend the stomach from the abdominal wall. Now, what they're going to do is they're going to stretch. They're going to allow the stomach to stretch. They're going to allow the intestines to undergo strenuous contractions and allow all of these. You've got about 18 feet of small intestine in your gut. And all of it now is able to move quite freely because it's bound up by these mesenteries. So it's going to hold everything in its proper place. So in this picture, not exactly something I would ask you to label, but what APR is trying to show you is that sort of in between all of the small intestine, there are these mesenteries. Again, as I said, they're helping to prevent your intestines from getting twisted and tangled. So you can do some cartwheels, and you're going to not have your intestines get all twisted and tangled like happens in horses, right? So what happens in horses? They roll, they get what we call colic, right? Their intestines get wound up, and they can die from that. So we don't have, I'm not exactly sure why horses are more prone to that, but I just know that they are. So you don't hear about us getting our intestines wound up and knotted up like horses do. Now, these layers of mesenteries are also going to provide a layer for blood vessels and nerves to travel, all these nerve endings, the enteric nervous system, and blood vessels to travel down to the digestive tract, and also a place where lymph nodes and lymphatic vessels travel. So again, you recognize the APR images, and you already know about the peritoneum being the layer around all the gut organs, the visceral being shrink wrapped to the organs itself, the parietal lining the abdominal pelvic cavity. And that's really all this is showing. I'll come back to the omentum in a moment. So what is the parietal peritoneum? Pariet means what? Pariet means wall, right? So this is the lining on the wall, on the cavity of the abdominal pelvic cavity. And it forms sort of a two-layered membrane around the digestive tract. Again, we call it the serosa, the outer layer. And some of this kind of hangs around within. I won't worry about the dorsal versus the ventral mesentery. There is the lesser omentum. The lesser omentum is some of this mesentery that extends from the lesser curvature of the stomach to the liver. And then the greater omentum is basically this big sheet that hangs from the greater curvature of the stomach, and it covers over the small intestines like an apron. And we didn't see it too much in the fetal pigs, but if you were to look at a human dissection, you would see sort of this big sheet, if you will, the greater omentum that goes around the stomach and covers over the intestines. So here's the lesser and the greater omentum. The lesser at the top, again, we saw the stomach and the liver, really, relationship there. And then the greater omentum is like this big apron sheet that goes from the stomach and hangs down over the intestines. A couple of terms here. The mesocolon, extension of the mesentery that anchors the colon to the back of the abdominal wall. So the mesocolon, mes, middle, it's going to anchor your colon. When I say colon, what am I referring to? Large intestine, right? It's going to anchor your large intestine back to the posterior abdominal wall. And some of your organs are intraperitoneal. So what does intra mean? Within, right? The peritoneum. So these are organs that are completely surrounded by the mesentery, completely surrounded by the serous membrane. This is going to include your intestines, okay? But there are other organs that are retroperitoneal. Now, when you hear the word retroperitoneal, you usually think of the kidneys. They are, in fact, retroperitoneal. The bladder is retroperitoneal. But when it comes to the digestive system, there is a portion of the duodenum, a portion of the pancreas, and a portion of the large intestine, which is actually considered to be outside of the peritoneal cavity. So it is therefore retro, at least in part, retroperitoneal. So looking at this picture again, I like this one better than the APR image. The artist rendering is nice. So the lesser omentum, right, this sheet that's connecting what? The liver to the stomach. And the greater omentum from the greater curvature, this is called the greater curvature of the stomach, going down, wrapping over all of the intestines. Now, that if we lift up the greater momentum, or greater omentum, lift it up, retract it, lift up that apron, we see, of course, the transverse colon. The mesocolon, what was that again? The mesocolon was what connected the colon back to the abdominal wall, so imagine it's being anchored back there. The mesentery in between, right, the layers in between. And much of what's going on in the digestive system is, well, the whole digestive system is about what? It's all about absorption of nutrients. We want the absorption of nutrients, we want the whole process to go slow enough to make sure we're able to absorb the nutrients. We want it to go fast enough, however, not to let the stuff rot too long, right? We don't want this stuff to rot. So we have a very highly controlled movement. It's being controlled by the nervous system, this enteric nervous system. It's being controlled by hormones, hormones released by your stomach, hormones released by your pancreas are helping also to regulate this motility. So you've got the nervous system and the endocrine system coming together to control the digestive system. So again, we have to control this very nicely. And what does the word pericrine mean? Neural, right? Nervous system, hormonal, endocrine. What does pericrine mean? Para means next to. So this is local control, if you will. So cells, maybe cells from the liver are influencing the stomach in a very kind of local neighborhood kind of way. So the neural control. I've already mentioned these two portions of the enteric nervous system. The short reflexes, the myenteric reflexes, these are going to be brought about by the myenteric plexus. And this is a stretch reflex or a chemical stimulation. And this is what's going to stimulate peristalsis of swallowing. And then there's the vagovagal reflex, or the long reflexes, and this is parasympathetic, and this is going to stimulate the motility and secretion of the rest of the digestive product. So short reflex, swallowing, long reflex, thinking about going through the rest of the longer portion of the digestive system. Hormones involved here, just going to mention a couple, gastrin and secretin. Okay, gastrin and secretin, they are released and travel through the blood and are influencing the digestive system. And then there are also chemicals released locally that diffuse to neighboring cells, and that's considered the pericrine. Not going into a lot of the details here on gastrin and secretin, I just wanted to say those words. You may hear about them in passing. Now, my notes here tell me to skip all the way over now to slide 98. So I'm skipping over the teeth and the salivary glands. Again, that's some of that bonus material. What I would recommend is that you just read through it once, and if you have more time, great, but at least walk through these slides once. So I'm going to slip all the way down, go past the teeth, go past the saliva and the salivary glands, although they're, I think, quite interesting, and go down through the entire swallowing reflex, through the stomach and the histology of the stomach, some of the hydrochloric acid and the pepsin and some of the enzymes made by the stomach. There's a lot of really cool stuff in there. If I have time, I'll go back and discuss a little bit more of it. But in the past, I haven't had time for this. And that brings us to the liver, right? Page 99, is that what I said? Am I being true to my notes? Just want to go back. Or did I skipped over from? Way back, right? What did I say in my little red note there on this page? Slide 98, right? So I'm going over to 98, which is the liver. So I want to pick up with the liver and the pancreas and the gallbladder and spend a couple of minutes on these important organs. So again, go back through that. Don't spend time looking at the major details. The questions I will ask you will be more global. So read through those slides for bonus. So let's start off with what's going on in the overall digestive process to get us to the liver and the gallbladder and the pancreas. So you've just swallowed some food. It's gone down into your stomach. It's been churned around by the 3 layers of muscle. There's hydrochloric acid down there. It's been mechanically and chemically digested. The food is going to leave the stomach as that pasty substance called chyme. and it's going to be dropped into the duodenum. Right? Picture that so far? What's the problem with acid reflux? What's the problem with acid reflux? The stomach is very acidic, hydrochloric acid, pH of 1 or so. If that acid gurgles up into our esophagus, what happens? Burns, erosion, real major problems for the esophagus, right? Over time can be esophageal cancer and a lot of problems. It's not much different for the duodenum. The stomach contents have been broken down by the acid, so what you're squirting into the duodenum is also quite acidic and could be rather damaging to the intestines. So what's going to happen is that as soon as things are going through that pyloric sphincter and are entering into the duodenum, we have to neutralize that acid. And the pancreas is going to be very important for neutralizing the acid. So the pancreas will be releasing bicarbonate. Now bicarbonate is a buffer, right? And it's going to help neutralize the acid from the stomach. So do you see, can you picture that point, right where the chyme is entering into the duodenum? And what is right there on our digestive models? Within just a couple of centimeters of where the chyme enters into the duodenum, there was the entry of the gallbladder, the liver, and the pancreas at a structure that Amerman called the hepatopancreatic ampulla, and that I also called the duodenal papilla. And remember, that's where you have the pancreas and the gallbladder and the bile and the liver secretions coming into the duodenum. That pancreas, right there, is releasing bicarb and neutralizing the acid. So that's a very important thing that's going on right there. And that junction, right there, where the stomach is dumping into the duodenum, is where the liver and the pancreas are all coming together. Everybody picture that moment, that place? So let's talk about the liver a little bit. The liver is your largest glandular organ. Weighs about 3 pounds, pretty big. We know it's kind of reddish brown. Reddish because there's a lot of blood flow, right? Your blood is being filtered largely through the liver. And there's also a lot of reddish brown color because there's also a lot of iron here. So you want to up your iron levels a little bit, have some liver and onions. Now, when it comes to the liver, there are lots and lots of functions, but the one we want to talk about here is the release of bile. So the bile is released by the liver, comes down and is stored in that gallbladder that we know about, and then is released when needed, and bile contributes overall to the digestion of fats, emulsifies them, breaks them down. The anatomy of the liver we never discussed before. The anatomy includes 4 lobes, 4 sections. There's the right and the left, the quadrate and the caudate. Well, you tell me, right and left, caudate must be toward the caudal, tail, or a tail-like structure of the liver. And quadrate, we'll see that in a moment. Now there's a ligament. If you ever look at any model of the liver or look at the liver, you're going to see there's a very strong separation, like almost like looks like a tendon, almost like structure, that separates the left from the right. And that is a ligament. It's a sheet of mesentery that suspends the liver from the diaphragm. So it really is sort of holding it up and separating it. It binds up to the bottom of the diaphragm. Then there's also a umbilical ligament. This is actually a leftover from the umbilical vein, and we'll see that that umbilical vein is carrying blood from the umbilical cord to the liver of the fetus, no longer needed after birth. The quadrate lobe we're going to see is right next to the gallbladder, and the caudate lobe is that tail-like lobe of the liver. So looking at them, pretty obvious, I think, which one is which. If you just remember that the caudate has this tail-like structure. So let's take a look right side, right, anatomical direction, left side. There's that ligament that's pretty strong, kind of divides it right in half. Then what do we have here? The quadrate, I told you, is where? By the gallbladder okay, down here by the gallbladder. And then the caudate, there's sort of like this tail-like structure, also more posterior. So there's that falciform ligament. It's in green. The round ligament is the end of it that you're seeing here. And of course, we recognize the big gallbladder sitting here as well. So what would this be? Right? Right? And left. What is the kidney, what is the liver doing? It's releasing bile. What's another major function of the liver? Filtering, right? Filtering blood. And you already saw in passing, when we were talking about capillaries, that the liver is full of sinusoidal spaces and lots of fenestrations and discontinuous capillaries where blood is exchanged easily. Now, how does blood get to the liver? Now, there's two routes of blood going to the liver. There's the hepatic portal vein. Portal, so somewhere in this system we're talking about a double capillary layer. In fact, the hepatic portal vein is bringing blood from where to where? From the gut to the liver. The blood was already, the nutrients were already absorbed at the capillaries in the intestines and then travel to another capillary bed in the liver. That's why it's called the portal. So we're going to a second capillary bed. And then there's also a proper hepatic artery. So which one's bringing oxygen to the liver? The hepatic artery. Which one is bringing the blood to be filtered by the liver? Hepatic portal vein. Okay, so one's bringing in blue blood coming in from the gut. You're going to filter through this stuff before it enters into the rest of the body. The gallbladder sitting there on the inferior surface of the liver, and there is what we call the bare area. That is the superior surface on the liver where it attaches up to the diaphragm. So the bare area would be where? Kind of up here, right? And we're looking here at the gallbladder. We got that. So let's take a look at this. We've got coming in. Can we see coming in? Yeah, we can see coming in. So the hepatic portal vein is coming in right here. Blue, right? Blue blood. And then there's also the, then there's also the proper or the regular hepatic artery. And they're coming in right here, kind of right next to the gallbladder. And then this large vessel leaving is what? That is going to be the inferior vena cava, and there's going to be a hepatic vein that we'll see in a moment that's going to carry blood out of the liver to the inferior vena cava and back up to the heart. Now, if you were to look inside the liver, what you would see are these sinusoidal structures, all these sort of hepatocytes. So these are all hepatocytes. These are the cells of the liver. And you'll see what looks like these hexagonal units within the liver. In the very center of all these hexagonal structures is a central vein. So blood is coming up through that central vein. And then at each of the apices, each point of this, you can't always see them everywhere, but at each point of the six-sided hexagon, there's going to be a triad, and that triad, you see a red, a blue, and a green. The green is a bile ductule. It's going to be carrying green bile. The red is the artery, and the blue is a branch of the hepatic vein. So what you see here is you have an exchange going on within the liver of blood coming up from the gut. Lots of exchange within these big sinusoidal areas. You've got bile coming through here, and you have the regular blood supply coming into the liver. And all of these sinusoidal regions are also going to be a place where there are many macrophages. And what do macrophages do? Eat, right? They gobble up things that could be harmful to you. Remember, the liver is breaking down your red blood cells. It's also breaking down things that could be harmful to you and toxins that come into the body. So when you look at the liver, when you look at that picture, you see these hepatic lobules. They are these cylinder-like structures. They're filling up the majority of the liver. In the center of them is the central vein. The hepatocytes are the cells. They're cuboidal-shaped cells that are around the central vein. And then you'll see these big spaces called the hepatic sinusoids. And they're blood-filled chambers, and they're lined by fenestrated endothelium that allows for a lot of exchange going on in this area of the liver. This is going to allow for a interaction of the blood coming from the stomach and the intestines. Sorry. It's going to allow for an interaction of the blood coming in from the intestines and from the stomach, and allow for a bunch of macrophages to be gobbling up any bacteria or debris that could be coming into your intestines that could be harmful to you. The name for these macrophages, there's a specialized name for them, and they're called the Kupffer cells. So the Kupffer cells, specialized name for the macrophages of the liver. And you shouldn't be surprised that these hepatocytes also have significant microvilli to increase the surface area, because so much about filtering is going to be involved with increasing the surface area. So as you eat, last slide here, as you eat, what do? After a meal, the hepatocytes are absorbing from the blood, right? So where they get it, the blood's coming in from your gut, being absorbed through the submucosa, going up through the hepatic portal vein, going to your liver. The liver is going to then, from your blood, absorb nutrients, glucose, amino acids, iron, vitamins, all that stuff. It's going to remove and degrade anything that could be harmful to you. But in that cycle, it could also be breaking down drugs. It could be breaking down toxins, breaking down hormones. That's a normal process, right? We have a half-life for all of our molecules. So our molecules are being broken down. It's also going to secrete into your blood what you know to be your albumins, the clotting factors found in your blood. We'll talk about angiotensinogen in the next chapter, and many other things. And between meals, right, so the liver is collecting glucose, and what is it going to be doing with that glucose? The liver is going to make glycogen. Glycogen is a storage molecule of glucose. And then between meals, when your blood sugar starts to lower down, your liver is then going to release that glycogen to maintain your blood sugar. Okay, so we have lots of things going on at the liver. So we'll stop there, and what I'll do is I will record a few more minutes on this. So I'm going to continue this on my own, and then I'll post this lecture on YouTube. And that will allow me on Thursday to start with respiratory part 1, next Tuesday to do the second part of the respiratory system and go through that material with great comfort. And then we have our exam a week from this coming Thursday. That exam is going to cover arteries and veins and blood pressure, vessels, chapter 20, chapter 22 that I'm covering right now, the highlights of the digestive system, and then the respiratory system, chapter 25. So continuing on with the liver, this is the rest of this conversation will be pre-recorded. It was not covered in lecture. Again, I'll be discussing some of the slides, and that material will be directly on the exam. The material that is bonus will be indicated on the slides. I'll be skipping over it. If you'll read through it, I'll ask you some questions on the next exam, and those will be bonus points. So within the liver, as we saw a moment ago, you have, think about what happens. So you're digesting food, you're absorbing those nutrients into the bloodstream. Those are traveling to the liver and are meeting into the liver through the hepatic portal vein. And from that, the hepatocytes, the cells of the liver, are going to absorb from your blood the glucose, amino acids, iron, vitamins, many other nutrients. They're going to be removing hormones and and toxins, detoxifying things, breaking down drugs and medications. They're going to be releasing into the bloodstream your albumins, clotting factors, things like that. And then between your meals, when your blood sugar begins to drop, the liver will break down some of that stored glycogen and release that glucose into the bloodstream to help maintain your blood glucose levels. Now, as you've seen, the hepatic lobules, these sections of the liver, are separated by what's called a stroma, a connective tissue layer. And in here, again, you have a lot of overlapping of the blood coming in and then leaving the nutrients, those macrophages, those Kupffer cells. And what you'll see is that there is a hepatic triad. The hepatic triad is made up of two vessels, a portion of the hepatic artery, a portion of the portal vein, as well as the bile ductal. And you'll see them, little triads, in all of the cartoon images. So in this APR image, what you're seeing here, go with black. So there's your central canal. That was in the very center of each of those. Remember, they're kind of hexagonal. You can't see the hexagonal section, but that's sort of that part of the hexagonal unit. And you have all these sinusoidal regions in here. At the edges, which you can't see here, would be the triad. So there would be the bile ductule, the branch of the hepatic portal vein, and the hepatic artery. And that's where all the exchange is happening when the liver is detoxifying and filtering everything. So again, you have a small branch of the blood coming in, you have a small branch of the hepatic portal vein, and both of those blood supplies are going to mix here in the liver. So you have incoming oxygenated blood filtering with nutrients coming in from the portal vein. And eventually, all this blood is going to be filtered through these sinusoids, a lot of those macrophages there to destroy things. And all of this blood with its nutrients is now going to leave the liver through the hepatic veins, left and right, which will then merge into the inferior vena cava. So you can imagine in this image, again, you're seeing that hexagonal shape. Here's an edge to it, edge to it. It's never exactly perfect, right? And there's the central vein. And over here we see this triad, the hepatic portal vein, the bile ductule, and the hepatic artery. I would not ask you to recognize those, but you'll see that they're running together as a triad. And then that blood is mixing and then is moving toward the central vein, which then comes back out of the liver and goes to the inferior vena cava. In the midst of all this, not only do you have the mixing of the blood, but you also have bile, which is made by the liver and is passing as part of this triad, and eventually will go to the hepatic ducts. And that will lead now the bile down the cystic duct. The cystic duct is the one joining down to the gallbladder. And again, this is going to be released when you need more of the bile. Also near the duodenum, and I mentioned this earlier, near the duodenum is where the bile duct is going to join the pancreatic duct. And it's there that we have the hepatopancreatic ampulla, or the folds of tissue there are also referred to as the duodenal papilla. So I'm referring to those as being essentially the same. The chamber is the ampulla, the fold of tissue, the nipple-shaped structure is the duodenal papilla, but I'm not going to differentiate between those two. You can think of them both being essentially the same thing. Now, the duodenal papilla is going to release bile under the control of the hepatopancreatic sphincter. The other name for that is the sphincter of Oddi. And that is going to release the bile and the pancreatic juices into the duodenum. Again, remember that the chyme coming into, the chyme coming into the duodenum has a very low pH coming from the stomach. And that must be neutralized. And part of that is the pancreas is releasing some bicarb, some neutralizing agents, which will bring that pH back up closer to neutral so it doesn't damage the duodenum. So you can see that here in this image. So here is the duodenum. Right up here will be the stomach, and we just drop through the pyloric sphincter. We're in the duodenum right here, not very far after that. There is that hepatopancreatic ampulla. The fold of skin is the duodenal papilla. And what's coming in there now are the pancreatic juices coming from the pancreas, meeting the bile coming down the bile duct from the gallbladder and from the liver. All that's coming down and dumping into the duodenum. Let's move on to the gallbladder. It's related, of course, to the liver because it's that little pear-shaped sac up underneath the liver, and it is going to not only store the bile, but it's also going to concentrate that bile by about a factor of 20-fold. So not only, again, is it storing, but it's also concentrating the bile, and it is going to release that bile as needed from the body. Bile itself is sort of a yellowish green fluid. It has a lot of things in it, but the portion I want you to know is that there are bile acids or bile salts that you'll hear about. These are basically phospholipid-like molecules that are going to help to emulsify or break down, to dissolve the fats from the diet. There's also going to be bilirubin, which is a pigment derived from the breakdown of hemoglobin. Remember that your liver is helping to break down the red blood cells. So as red blood cells are recycled, they only live for 120 days. As they're broken down, the heme group is going to be released, and in the breakdown of that heme group, the bilirubin is a part of that. That's what's going to also give your brown color to your feces, because now this urobilinogen is going to be released into your duodenum, go through your gut, and be released as part of your feces. If the bile becomes excessively concentrated, then you can have the formation of gallstones. That usually is going to cause the gallbladder to be removed. Imagine a gallstone would be blocking those ducts, and you can live quite well without a gallbladder. Now, the liver is secreting about 500 to 1,000 milliliters of bile every day. I mean, that's a huge amount, but remember, most of it is some of it's being reabsorbed, and then the gallbladder is concentrating it such that much of it is returned through your ileum and to the liver itself, and then some of the bile acids are also being released in the feces. You have to have cholesterol to make more bile. So bile, again, is a cholesterol derivative, another reason why we must have cholesterol as part of our diet. Again, if there is a buildup of too much of a concentration of the bile, or if there's, just like in kidney stones, too much of a concentration of calcium carbonate, okay, calcium carbonate, and or bilirubin, all of those things as they become concentrated can form the calculi. Remember, calculus is the hard stuff on your teeth. Most of you might say that calculus is a pretty hard math class. So just remember, when you see calculi, think hard. And this is going to form gallstones, and gallstones are more often seen in women over the age of 40. Typically, you'll see more often in women with obesity because, again, they have excessive cholesterol in their bloodstream and in their body, and that is going to lead to the bile stones. If you have those ducts obstructed, it's an extremely painful process. Also, you can become jaundice because you're also interfering with liver function to a point, and the skin can become sort of yellow, like cirrhotic changes. You also may have problems after a gallbladder attack or after having the gallbladder removed, an issue with the absorption of fat-soluble vitamins. Again, you're not making as much bile. Yes, you're constantly making it, but you're not concentrating it quite the same way with the gallbladder. And so some individuals will talk about having sort of a digestive issue after a fatty meal, or may see problems with absorption of their fat-soluble vitamins. Remember, those fat-soluble vitamins are A, D, E, and K. To get rid of stones, you might be put under lithotripsy. Lithotripsy, remember, lith means stone, right? So you're basically sonicating, vibrating with ultrasonic waves, those stones, helping them to pass without surgery. If that's not successful, then the gallbladder would be removed. Okay, now the last organ here is the pancreas to talk about. You know that it is a spongy organ. It's when you saw in the fetal pigs in 105, many students had a hard time locating it. It is a very glandular, not a very easy organ to see sometimes in the fetal pigs. It is posterior to the great curvature of the stomach. It is sort of near the duodenum. You saw that it's draining into the duodenum. There's the major portion called the body, and then there's a tail that tails off to the left. Recall the pancreas is both endocrine and exocrine. Endocrine because it is making hormones, things like insulin and glucagon. But it's also, an even bigger part of the pancreas is making pancreatic juices. And these pancreatic juices contain within them a lot of digestive enzymes, as well as those neutralizing compounds like bicarb that are going to neutralize the stomach acid as the chyme enters into the duodenum. The pancreatic duct is traveling along the middle of the gland and again dumps in with the bile duct at the hepatopancreatic ampulla and is released into the duodenum. There's actually a second smaller duct called the accessory pancreatic duct. It branches off from the side of it. It independently distributes or releases pancreatic juice into the duodenum even when bile is not. Again, your body needs bile specifically when you're eating a fatty meal. It doesn't need bile all the time, but it does constantly need a dripping of the pancreatic juices to neutralize the chyme and for digestion. And so we need to have a second route, and that is provided by the pancreas, by this accessory gland. Again, the pancreatic juices are quite alkaline, and that is going to help to neutralize the acid coming from the chyme. There are lots of enzymes involved with digestion, and the sodium bicarb is a big part of it. If you looked at a pancreas, it looks really no different than a salivary gland or a tear gland in that you have those acinar cells. They're releasing their products into a duct. Remember, this is going to be exocrine because these digestive juices are being dumped into the intestines, which are then have access to the outside world. So these are exocrine glands within the pancreas. I wouldn't ask you to recognize anything here, but you can see that the pancreas is made up of many of these acinar collections. Again, those are the structures that are making all of the products released through ducts into the duodenum. A couple of key enzymes. Now, enzymes that are released in an inactive or preform are called zymogens. And there are many of your enzymes are released as zymogens. That basically means that they're released in an inactive form, which then become activated after they are released. Trypsinogen is one of those. And it is released by the pancreas, a very, very important enzyme in the breaking down of your proteins. And trypsin and chymotrypsin and this procarboxypeptidase. They're all important enzymes. I'm not going to get into exactly what each one does, so don't worry about that. But what you will see is that on some of these, there's the ACE ending suggesting that they are enzymes. There's amylase that breaks down starch. There are lipases that break down fats. And there are ribonucleases and deoxyribonucleases that break down RNA and DNA. So again, what do you got everything, right? You've got proteases, the trypsin breaks down proteins, sugars, fats, and nucleic acids. So the pancreas is able to basically digest, break down any sort of macromolecules that you bring into your body. That's what's shown here, basically, that the pancreas is releasing. There's your pancreatic duct. Comes in, there's your major, okay, and there's a minor that we can't see here that comes around the back, the accessory duct, and then those proenzymes or those zymogens are released, and eventually they make all the other enzymes necessary. Remember, the stomach was the site of breakdown, right? Breaking down the food, mechanical digestion. That chyme was released into the duodenum, and it's here now. You're going to start to continue to break it down and to absorb those nutrients. Those nutrients will be absorbed into vessels, carried off to the liver, right? We talked about this already, through the hepatic portal vein, and then into the liver, into the sinusoids, mix with the arterial blood, get those Kupffer cells to detoxify, break things down. That blood is then returned back to the circulatory system through the hepatic vein, which dumps into the inferior vena cava. Lastly, when it comes to what is it that stimulates the release of these pancreatic juices and the bile, there are three stimuli that are going to cause the pancreas and the bile to be released. Number one, acetylcholine. Well, when you hear acetylcholine, a lot of things may come to mind, but acetylcholine is the neurotransmitter that is going to be released from the vagus nerve and those enteric nerves. Remember I mentioned the enteric nervous system, all those nerves of your gut. Well, they release acetylcholine when they're being stretched and activated, and this is going to cause now the release of these enzymes to begin to break down the food. So acetylcholine's one. Number two, there is a substance called CCK, and CCK is released by the mucosa of the duodenum, and this is going to be released in response to the arrival of fat. So clearly when fats come into the small intestine, you better get bile involved and get bile released. And in addition, you're going to have other enzymes released as well. So it's really going to stimulate the gallbladder to squeeze and spit out some bile. And then finally, there's an enzyme called secretin. It's also released from the duodenum in response to acid. So as the stomach is dumping its products into the duodenum, it's quite acidic. Those cells now in the duodenum are going to release secretin, and that basically tells the pancreas, hey, you better release some of the bicarb, some of that alkaline fluid, to raise the pH and neutralize the environment. Now I'm going to skip over the conversation over the small intestine. Again, most of this will be anatomy review. Read through this, and I believe you will be just fine for some extra credit questions on the next exam. And then that brings us to a few things about chemical digestion. So I want you to appreciate where in the digestive system different macromolecules are broken down. So we'll start off with carbohydrates. Starch is the most digestible carbohydrate, and cellulose is indigestible. So starch, right? We get starch from plants, we get cellulose from plants. Starch are the things that we can break down. The cellulose is the roughage that we can't break down, like in hay. Now, some animals, like cows, can break down cellulose, but we don't have those enzymes. And so if we were to eat hay, it would pass right through us. Now, the starch is a long chain of glucose molecules. So eventually, as starches are broken down, we are able to release glucose. Now, the carbohydrates, starches, first start getting broken down in the oral cavity by amylase. Amylase is the enzyme that breaks down starch, and there are amylases released by your salivary glands. This particular kind of amylase works really well at neutral pH, which is what you find in your oral cavity, which is why when you eat a saltine, right, eat a cracker, which is starchy, you can get a sweet sensation from that because immediately you're breaking down that starch into glucose. Now, in addition, there is additional amylase released by the pancreas. So think about it. Amylase only works around neutral pH. When the salivary amylase reaches the stomach and we're down at a pH less than 4.5, the amylase no longer works. So only about half of your carbs are actually broken down when they reach the stomach. The rest of those starches then will come through the stomach undigested and reach the duodenum. And here the pancreas will also release additional amylases. Remember, here we've also neutralized that pH. And so the pancreatic amylase will continue to break down the additional carbs to make the glucose molecules that are then absorbed into the intestine. Now, all of this is happening. The pancreatic amylase, again, is going to convert the rest of the starch into simple sugars, and those simple sugars will be further broken down by enzymes found in the brush border. The brush border is the microvilli mucosal layer of the intestines, and again, that will cause the breakdown of the sugars into their smallest part. Now, some people start having problems with the breakdown of lactose. Lactose is milk sugar. People who are lactose intolerant, what happens is that they no longer make an enzyme called lactase. Lactase breaks down lactose, and so if you are not able to break down lactose, then that sugar is remaining in your gut, and the bacteria in your gut can start breaking it down, and that can lead to some gassy side effects. So people who are lactose intolerant will complain about gas and some issues following a consumption of lactose. So now we give, right, the person takes a Lactaid, basically a pill that has lactase in it that breaks down the lactose for them. Now, carbohydrates again are being absorbed into the gut. I'm not going to get into all of the specific steps involved with the uptake of glucose, but just know that most sugar absorbed into your gut is going to be this glucose. It is brought up into your gut by facilitated diffusion, and then that's going to then drag and go into the blood vessels. So we'll skip over some of the specifics here, but what I want you to realize is that those sugar molecules, once they are absorbed into the intestine, are going to make their way to the liver via the hepatic portal vein system. Again, as I mentioned, those people who become lactose intolerant, the lactose gets all the way down, doesn't get digested, and that's where bacteria in the gut start breaking down that lactose, and that can lead to some rather uncomfortable side effects. This is more common in some individuals. So nearly all Asians are lactose intolerant. A large percentage of Mediterraneans, a very high percentage of African Americans are lactose intolerant. It's less of a problem in American whites, and again, it's just an issue with genetics and whether or not you have that. Now, if you consume yogurt and cheese, that's usually not as much of a problem for individuals because there are bacteria in those products that have already broken down the lactose, so there really isn't as much of the lactose coming in. So that's the quick story on sugars. Now, proteins are again made up of amino acids, we know that, and those amino acids don't start getting broken down in the oral cavity. So that's one of the big things, is that the proteins that you're bringing into your body don't start getting broken down until they get down into the gut. we're bringing in quite a bit of proteins every single day into our body. The enzymes that are going to break down proteins are going to be some sort of protease. You may see them called peptidases as well. And they start working at a lower pH of the stomach in that 1.5 to 3.5 range. One of them is pepsin. Okay, pepsin breaks down proteases, and many of the other enzymes like trypsin and chymotrypsin that I've already mentioned are also continuing to break down proteins in the small intestine after they've gotten those enzymes from the pancreas. So we won't worry about the individual enzymes here. Just know that there are a whole bunch of enzymes, carboxypeptidase, aminopeptidase, dipeptidases, and they're all breaking down amino acids and proteins into their smaller parts. So again, we're just breaking down into smaller individual amino acids, and then those individual amino acids become the building blocks for other proteins in our anabolism. Again, amino acids are going to be taken up through the cells of your intestines by the process of penocytosis or absorption into those cells. Again, we won't worry about all the details here, just the big idea. When we get to lipids, remember lipids are hydrophobic, which means that their absorption is going to be more complicated, and the enzymes that are going to basically break down lipids are going to be any sort of lipase. Now, there are lingual lipases, that is, lipases secreted in your salivary glands. So they're active in your mouth, but they're even more active when they get down to your stomach. So a small amount of fats start getting dissolved from your oral cavity. The pancreas is also secreting a large amount of lipase, and that fat is entering into the duodenum. Again, that was one of those clues, right, that the bile needed to be released. And what happens is that that bile will now emulsify or help to dissolve, if you will, the fats. Lecithin and bile acids, you can buy lecithin in the drugstore. Lecithin is actually going to help some people with their absorption of fats, the breakdown of their fats. So the intestines are going to be moving products through via peristalsis, and then there's also going to be segmentation. As the name suggests, this is when a section of the intestines is going to basically segment itself off, and then there'll be a lot of churning and agitation in that area, and that's going to really help coat those fats with the lecithin and with the bile acids. And basically, just like making mayonnaise, you have to emulsify the fats into something where they'll sort of dissolve. Now, one of the major lipases acts on triglycerides. Remember, triglycerides are the fats, some of which are traveling in our blood, and we want to minimize those fatty acids in our blood. So fatty acids, then, you've got a big old globule of fat. We're going to emulsify it with things like lecithin and bile salts, and that's going to coat them. That's why you need that segmentation in the gut, and that's going to allow them now to be carried through the body. Now, these lipids are going to be these little minute droplets, and this is going to allow us now to absorb these molecules into the body. Again, there's more details on here than I'm going to hold you for. Just the overall idea of how carbs, proteins, and lipids are absorbed and broken down. Again, those fats are going to pass through the duodenum and be transported. And then, going back, it turns out that lipids are not going to be absorbed directly into your intestinal cells, but are also picked up by your lacteals, part of your lymphatic system. So we won't worry about the specifics on this slide. Okay, all I want you to realize here is that fats are going to be taken up by these lacteals. Lacteals are these lymphatic structures lining your intestines, and they have larger openings, if you will, than your blood vessels. And so the larger fat, these little micelles, these clusters of bile acids, lecithin, and fats are going to be absorbed into the lymphatic vessels. And then eventually they will re-enter your bloodstream, but they're not going to be directly going to the hepatic portal vein to the liver like other nutrients. So don't worry about the chylomicrons. We'll just skip over this slide completely, no concern. Finally, nucleic acids. We certainly do consume nucleic acids. It's not something on our nutrition labels because we don't get any direct energy from them. But we certainly do have from our pancreas nucleases made. These nucleases, again, are going to break down DNA and RNA that we consume. Every time we're eating any kind of cell, we're going to be consuming and breaking down the DNA and RNA of that organism. Vitamins are also absorbed directly. The fat-soluble vitamins, A, D, E, and K, are going to be absorbed with other lipids. Water-soluble vitamins like B and C are going to be absorbed directly in by simple diffusion into your bloodstream. Minerals are also absorbed along the small intestine, things like sodium, chloride, magnesium, all being brought in. Iron and calcium will be absorbed as needed. We mentioned that whole story with the iron earlier when we were discussing overall iron metabolism. Calcium comes in through active transport. So this is the first time I think I've mentioned anything about active transport. So it costs energy for the body to absorb that calcium. The exact process by which it happens, we won't concern ourselves with. Now, why is it that the body is going to decide to increase its absorption of calcium? It would be under hormonal control. We already know about PTH. We know parathyroid hormone is released in response to low blood calcium levels. That is going to then upregulate your absorption of calcium into your body. Water is also being constantly brought in. We talked about water in the first lecture, and every day your digestive tract is receiving somewhere around 9 or so liters of water. And we already know the sources of that water is coming in through your food, your drink, and through a large part through your gastrointestinal secretions. Much of that is reabsorbed. Those fluids are reabsorbed along the small intestine and large intestine, such that with your feces, you're only losing a couple hundred milliliters every day. Now, we want water to be absorbed at the proper level. Obviously, if we have too much, when water, when the large intestine absorbs too little water, right? Too little water, then when the large intestine absorbs too little water, remember that the large intestine is not involved with absorbing nutrients, but instead is pulling all the last little bit of water out of the feces. So if the feces stay too, quote, wet, right? The feces stay too wet, then you can have diarrhea. And constipation would be the opposite, where now there's too much water getting reabsorbed, the feces become too hard, and they don't pass through the GI tract very well at all. The last thing we're going to do, which I think is a slight variation on what you have in your slides, I'm going to jump all the way over to 198, and I'm just going to talk about the last 4 slides or so. Please look over the large intestine on your own. And I just want to go over to the last little bit, just because I think it's great facts to know. So over here, when you get to microbiology also, you'll talk about flora, the normal bacteria found within your body. And it turns out that within your large intestine, there are over 800 different species of bacteria. Now, they're there to help break down nutrients. They also are very important in helping you make vitamins. So without some of the bacteria in your gut, you wouldn't be able to make, for example, vitamin K. Now, as a result of all these bacteria, We all deal with about 500 milliliters a day of gas, right? And actually, that's how much we actually produce that is going to leave our body, and that's just a fraction of the total gas that is present but is reabsorbed into our body. Most of that gas is going to be coming in from swallowed air, but also some nastier gases that are produced by the bacteria, and these are the gases that can give our flatus, our intestinal gas, a little bit of a lovely odor, right? And so hydrogen sulfide is a very stinky gas, and we've all experienced that from our family members at certain times. Now, it takes about 12 to 24 hours for the meal to be reduced through feces. And along the way, the body is breaking down and reabsorbing water and electrolytes. And so that means that, within 12 to 24 hours after eating a meal, your body has been able to utilize its nutrients and releases what it can't use. Your feces are actually about 75% water, 25% solids, and of those 25% solids, a good third of it is bacteria. Some are the undigested fiber, some of it's unused fat, and then of course you're going to have some mucus from along the mucous membrane and a lot of epithelial cells that were sloughed off throughout the entire process. Now, of course, the amount of fiber, the amount of fat would be variable based upon your diet. You know that each of the little sections of the large intestine is called a haustrum. And so those little sections of the colon are contracting about every 30 minutes, and that's going to keep the motility of your colon moving. And that's going to keep you moving products through by mass movement, such that you're having between 1 and 3 bowel movements every day. As the feces are moving through these haustral contractions, you will eventually get down to the rectum. The rectum will get stretched. There will be stretch receptors there that are going to stimulate a defecation reflex. This is that urge that you have to go. And this is going to be part of that myenteric plexus as well, giving you some weaker contractions. And then hopefully you will also have the complete control over this. Your parasympathetic nervous system is also involved with this defecation reflex, so your spinal cord is involved with this. Individuals with spinal cord injuries lose this reflex and so are not able to defecate on their own. Again, defecation occurs if the external sphincter is voluntarily relaxed, assuming you have control over that. You can also, with abdominal contractions, the Valsalva maneuver, help to increase the movement of defecation, and that's by increasing the abdominal pressure and basically lifting the anal canal up, and the feces will then move out of the body more easily. So the neural control of defecation is as follows. Again, this is great information to know at a dinner party. So the feces are filling the rectum. It's going to push on the internal sphincter, and then that's going to send signals up. So let's take a look at this. So feces are coming down the sigmoidal colon into the rectum. They're pushing against those stretch receptors. Those stretch receptors are now sending signals up to the spinal cord. Again, this is reflexive. It's going to send now parasympathetic signals down to relax those muscles, and also send voluntary motor fibers down. So you're going to be relaxing the internal sphincter, and then you're going to have voluntary control over the external sphincter, which is going to allow you to defecate. So we've learned about this in part long ago. A person was accidentally shot. This is, close to 200 years ago, and there was a large enough hole seen in the intestines in the stomach to better understand all of this. And this is where we finally understood that the stomach was making HCl, and all of this is If you go up to Mackinac Island, you can learn more about this because it was there that much of this research was understood. There's a little display up there on the island. And that does bring us to the end of the digestive system. So please go back, review the slides that I did not cover specifically. Those will be extra credit. And the things that I specifically discussed will be part of the exam. Again, the exam is going to be next week. It's going to cover the Chapter 20, Arteries and Veins and Blood Pressure. It's going to cover this digestive material and the respiratory system, which is coming up the rest of the week.